Okay, for assessment number 2 then, there's 4 main topics that we've looked at. These are the first 4 topics on paper 2 and paper 3, so year 13 topics. Now, investigating populations, there's 3 main methods that you must be aware of. Now, the first method is to investigate the population numbers of different species of plants in a given area. So that's going to be random sampling. Now, because plants do not move, we can use quadrats. So we're going to generate random coordinates using random number generators. You place the center of the quadrat at the random coordinates, and then we can do percentage cover or abundance for that given area. The second method is also to do with plant species or plant biodiversity. This time though, it's not in a given area, instead it's a line from A to B, so that's called a line transect. Now that second method is basically looking at the biodiversity in response to some environmental gradients. Now it might be increasing light intensity from the line A to B, or it could be the salinity of the soil, for example, from A to B, but that is plant species. And the third method is to do with animal species. Now, because animals do move around, we can't use quadrats anymore. So instead, we have to use the mark, release, and recapture method. So you guys need to go through that and the limitations of that experiment. Now, the second part to this test was succession. So these 2 booklets were actually put together this year. Now, you guys have to understand primary succession, and that's normally where you get a very hostile habitat, such as rock face. Now, you're going to start with a pioneer species. Now, what they do is they can colonize the harsh habitat, and they alter either the abiotic or the biotic factors within the environment. And you give an example of how they've done that. And then basically what happens is we get different species emerge over time, and they outcompete the previous species for some named resource. So the habitats become less hostile over time, and eventually we'll get to a climax community, which is a very stable community that does not change very much over time. Now, this question that you can see in a minute is actually about secondary succession. So the only difference there is that primary succession, there were no previous species present, like rock face, but secondary succession, we're going to get some sort of environmental disaster like a flood or a forest fire that's basically going to allow new species to start to emerge. So primary succession is on the spec, secondary isn't, but that will be an application AO2 question. Now, the third topic on the test is going to be photosynthesis. Now, we know producers can photosynthesize like plants and algae. Now, inside the chloroplasts, which are the organelles of photosynthesis, we get thylakoid membranes where we get the LDR reactions, and that's going to make ATP and NADPH. Now we know these 2 molecules are needed in the LIR reactions in the stroma, which is basically the Calvin cycle, and we're going to get carbon dioxide fixation onto RUBP, the 5-carbon, and you get 2 glycerate phosphates that are both reduced to triose phosphate, which are then used to make or build glucose or to regenerate the RUBP. And the last one then is respiration. So we've got aerobic respiration with oxygen, and that's the final electron acceptor inside the mitochondria to make H2O. If there's no oxygen present, we're going to get anaerobic respiration that only involves glycolysis, and we can either have ethanol and CO2 if it's plants or yeast, or if it's muscle cells, we're going to get lactate in the cytoplasm. But the mitochondria are not involved during anaerobic respiration where there's a lack of oxygen. Now, with question 1, it says ecologists studied a stream community before and after a flood. So technically this is secondary succession. Now, if it's a stream community, that means obviously we're going to get aquatic organisms. Now, I would start with the definition of community, just to remind yourself of what that means. So a community is all the individuals of all the different species present at a given time in that given area. So we're going to look at the change in the composition of the community over time, and that's called succession. So the table shows how the populations of 6 animal species found in the stream change following the flooding. So for me, this is really important because it says animal species, and that's twice there. It's also in the header. So we're not talking about, I could put here, not talking about plants. So we've got 6 species that we can number, but we are going to use the Latin binomial names with a capital for the genus and a lowercase for the species. We've got the number of days after flooding starting from day 1 and going up to day 63. So that's increase in time. And they've counted not only if the species is present, but also the numbers of organisms within that particular population of that species. Now this is meters cubed per meter cubed. So this is the volume, because we know it's an aquatic habitat, because we're told at the top it's the community in a stream. So we're not really going to be thinking about abiotic factors too much here. We know if we've got plant species that die and get decomposed, it can make the soil thicker, it can introduce more humus into the soil, more nitrates over time into the soil. We're going to be talking more about predator-prey relationships or variety of food types when we're talking about animal species. So let's start looking and annotating the data then. So one day after the flood, hopefully you guys can see that the species at the top and the species number 5, okay, we've got some of these individuals present. So there's actually 2 pioneer species, number 1 at the top and number 5, and you're obviously going to use the names within your answer. So you copy the Latin name carefully. And there's 0 individuals of the other species of R6. So the species richness at day 1 is 2 different species. Now, if you look at the species richness at day 63, hopefully you can see that there are individuals of all 6 species present. So the species richness at day 63 would be 6, which is an increase from 2 at day 1 after the flood. Now we know the pioneer species, they alter, in this case, the biotic factors within the habitats or within the environment. Now imagine these animal species are prey for other animals. So we can see actually at day number 5 after the flood, there's actually quite a big increase in the individual numbers of C. paradoxa, which is species number 4. So it might be that the 2 pioneer species are prey for this new species of animal that starts to increase in number. So we've had pioneer species alter a named factor in the environment. Maybe they supply or they are the food for other species to emerge. C. paradoxa increases in numbers from day 5 onwards, and it continues to increase in number. We can also see other species starting to emerge and increase in their numbers. So for example, L. packeri, species number 2, starts to increase from day 13 onwards. So species 2, L. packeri, and species 4, C. paradoxa, they actually have quite high numbers of individuals at day 63. Now, if these are dominant species that are going to increase in their population number, they're going to outcompete some of the other species present. So they might feed off these other species. So the numbers of the other species that were present, the previous species, would start to decrease. So let's look for a decrease. So if you look at species 3, we can see over time the numbers of species 3 are going down. We can also see that the numbers of species 5 are decreasing from day 22 onwards. So you want this idea of a change in the biotic factors, such as more prey being present. we get new species start to increase in number that can feed off or outcompete the previous species in some way. So we get a change in biodiversity, and in this case we get an increase in species richness over time. So not only do we get an increase in the total number of individuals, which would be the index of diversity with 6 species, we get an increase in the number of species from 2 to 6. Now, where it says succession, I would probably just write down a note to yourself that succession, when you define it, is the change in the number and type of species present over time, or the change in the composition of species present over time in that given area. So the answers then, if you guys want to go through them, the first mark point was there's an increase in species richness from day 1 to day 63 after the flood. Or you could say there's an increase in species diversity, or there's an increase in the total number of living organisms from day 1 to day 63. You could give the 2 Latin names of the pioneer species that are present at day 1. Then you give how they change, the example of how they change the environment. So they might be food for other species that will then start to emerge. You then name one of the species that starts to emerge and increase in population number. They can now, this is really important, colonize that environment in a particular habitat. So the habitats become less hostile over time. So new species can then colonize. And that is because of the change, or because of the increase in the pioneer species numbers. Now, remember, we're not allowed to talk about plants here. These are not plants. These are animals. You then need to give an example of how one of the species or more might be outcompeted. So they might be eaten as succession continues, or they might be outcompeted. So one example of where the species numbers go down, because we've had a nice example of where species numbers go up, and individual numbers within those species go up. Right, the populations of C. paradoxa and L. packeri both increased from day 13 to day 63. Now, that's a time period of 50 days. So day 13 up to 63 is 50 days. It says calculate how many times the population growth per day of C. paradoxa is greater than L. packeri between these days. So if we were to start and circle day 13 and day 63, okay, we can actually write on here that's 50 days. We go to L. packeri, and its value is 0.25 at day 13 and 29.5 at day 63. So if you go 29.5 minus 0.25 gives you the value over 50 days. And we can do the same for C. paradoxa. So 21.3 goes up to 182.6. So 182.6 minus 21.3 gives you that value over 50 days. Now, it does say growth per day. So we need to take these two values and divide both by 50 to get per day. So C. paradoxa was 161.3 divided by 50 days is 3.226 per day. L. packeri was 29.25, again divided by 50 is 0.585. And now we need how many times is this value bigger than the second value? So we go 3.226 divided by the smaller value will give us an answer of 5.5. So we can put 5.5. And that will get you 2 marks. Now, if you had a value of 3.226 and a value of 0.585, that will get you 1 mark for working out, even if you didn't have the correct answer in the answer box. Now, the stream eventually recovered to reach a climax community give two features of a climax community. Now, it says the stream, so we know it's an aquatic ecosystem, eventually recovered over time. So the values you've got at day 63 are not necessarily the climax community. We know there's two dominant species, but it might just be that if we had more recordings over a longer period of time, we'd see actually there's a stabilization of the population numbers with those particular species present. So a climax community we know is very stable. It does not, so very stable, it does not change over time very much. The species remain fairly constant. The population numbers of each species remain constant. And it's really only a change in abiotic factors like the flood that we had earlier that could alter the stability of the climax community. So very often you get dominant species that are present. So the first mark point was for saying that the same species are present over a long period of time. It's nice and stable. Population numbers for each of the different species also remain stable. They don't fluctuate very much. And abiotic factors remain constant, which allows the community to remain constant as well. If there's a flood, for example, that's a change in non-living factors. Could be a forest fire, which then could obviously affect the stability and reduce the climax community, reduce the number of species or reduce the population numbers. Now, this thing about carrying capacity then is really the maximum population of a given species that can be sustained by the habitat in which it lives. So carrying capacity of any population is the maximum population number possible or sustained by that particular habitat in which the species lives. Okay, question 2 then, it says the scientists investigated the effect of temperature and light intensity on the rate of photosynthesis in a species of plant called the creeping azalea. So they investigated the effect of temperature on the net rate of photosynthesis at 3 different light intensities. Okay, so the net rate of photosynthesis is what they measured, and they used high light intensity, and then they measured the net rate of photosynthesis, medium light intensity, and again they did the same, and they actually also investigated low light intensity, and they did the same. They also investigated the effect of temperature on the rate of respiration. So on the right hand side you've got the rate of respiration. Now this looks quite complicated, this data. However, if we break it down slowly, it makes it more accessible. So hopefully you guys can see that there's 2 y-axes, one on the left and one on the right, and the independent variable is the temperature, which sets on the x-axis on the bottom. So if we look at the dotted line first and that data, hopefully you guys can see that I've covered that in with a pink line. Now if you look at the key on the right hand side in the brackets, you can see the dotted line is the rate of respiration measured as the production of CO2 gas in milligrams, and that is per gram of plant per hour. So you should link the dotted line with the right hand y-axis, and it's the mean production of CO2 in milligrams, because we know CO2 is a waste gas from aerobic respiration, and that is per gram of plant per hour. Now the solid lines then, the data there, we've got 3 sets of data at different temperatures. We've got low light as one experiment at different temperatures. We've got medium light at the different temperatures and high light at the different temperatures. So we've got 3 separate experiments. Now the blue data represents the net rate of photosynthesis measured as the uptake of carbon dioxide, and that is in milligrams per gram of plant per hour. So here it's the CO2 uptake, not production, but uptake or fixation in milligrams per gram of plant per hour. So the CO2 we know is being fixed by Rubisco, and it's added to RUBP, which is a 5 carbon, and that's going to make 2 molecules of glycerate phosphate. So this is the Calvin cycle. And the first step of that is CO2 fixation, which is going to use up carbon dioxide. So the net rate of photosynthesis on the left. Now the question says below, name the factors. So factors means plural, so that means at least 2 factors that limits the rate of photosynthesis between point X and point Y. Now, any limiting factor then, if you increase that factor, you will also get an increase in the measured variable. So if we increase light and we had an increase in the rate of photosynthesis, the light must be a limiting factor. So if we look between points X and Y, and you should really get in the habit of annotating this data, we can see as low light increases to medium light, there's actually an increase in the net rate of photosynthesis. And also as the temperature increases, if we just pick one light level, for example, low light, and we increase the temperature at low light, we get an increase in the net rate of photosynthesis. So if you increase light, we increase the net rate of photosynthesis. So light must be limiting. And also, if we choose one light level and we increase the temperature, we also increase the net rate of photosynthesis. So we know if we increase light intensity, then we get a faster rate of photosynthesis because we get more LDR reactions. And actually, if we increase temperature, we increase the kinetic energy of molecules and substrates and the enzymes, and we get more enzyme substrate complexes, for example, in the Calvin cycle, which is the LIR, and therefore we're going to produce more glucose, which equates to a greater uptake of carbon dioxide. So temperature and light were both needed for that mark point. Then it says, use the info from the graph to explain your answer. So an increase in temperature causes an increase in the rate of photosynthesis. So the temperature must be limiting. Or you could have said increases the uptake of CO2 in milligrams. So that's mark point number 1. Right, mark point number 2, an increase in light or more light, or going from low light to medium light to high light causes an increase in the rate of photosynthesis or the net uptake of carbon dioxide in milligrams. So increasing one factor increases the rate of photosynthesis, and those two factors were both light and separately temperature. Now use the information from the graph to find the gross rate of photosynthesis at 20 degrees C and medium light intensity. So definitely we're going to highlight and circle gross, that means total. And 20 degrees C and medium light are also very, very important. So gross rate is basically the total rate of photosynthesis. Now, if you guys remember, we've done GPP, which is basically NPP plus respiration rate. So gross primary productivity, okay, is basically the conversion of light energy to glucose over time. NPP is basically the dry biomass of a plant, which is all the carbon molecules. And then obviously respiration is the rate at which glucose is used up. So that is not part of the dry biomass because some of that energy is being released as heat. So if we look at the data then and we use 20 degrees C, because it gives us that. So we're going to annotate 20 degrees C and draw a red line or a pencil line on the graph, and medium light. So we could say at medium light intensity, the net rate of photosynthesis is going to be 2.4. So you guys have to read that across very carefully. 2.4 milligrams of CO2. Okay, and that's being used up. But also, okay, we know that the respiration rate at 20 degrees C is the y-axis on the right-hand side, which is 0.35 here. If we read that carefully. So what we're going to do is add the net rate of photosynthesis to the rate of respiration to get the gross rate of photosynthesis. So gross rate equals the net rate plus respiration rate to get the total rate of photosynthesis. So we're going to take 2.4 and we're going to add that to 0.35, and then the answer was, if you add those two values together, 2.75 for 1 mark. Now they did accept a range of answers up to 2.81, but you should have got 2.75 or something slightly above that. Okay, creeping azalea is a plant which grows on mountains. Now scientists predict that in the area where this plant grows, the mean summer temperature is likely to rise from 20 to 30 degrees C. So the temperature is going to go up very slightly. Now that's quite specific, so we're going to use that on the graph. I'm going to have a look at that in a minute. And it also says the plant is likely to grow in this area that becomes more cloudy. So if it's more cloudy, then that is going to reduce the light intensity. So we're going to have low light intensity. Now this is one of the experiments that was done, of which the data is now on the graph. So more cloudy or lots of cloud, low light intensity, and we can use that data as well as the increase from 20 to 23. Now the question says, this is really important, describe and explain how these changes are likely to affect the growth of creeping azalea. So the first point is going to say, do these conditions either increase or decrease the growth? So you've got to select which one you think it is, and then you can explain it using the data from the graph. So let's have a look at the data. So 20 degrees C we've started at. Now we're going to increase the temperature to 23. So draw another line on your data. And we know it's medium light. Sorry, it's low light, isn't it? It's low light. So if we look at the low light value, we can see, hopefully if you guys do this carefully, that the net rate of photosynthesis decreases between 20 and 23 degrees C. So this in blue, this data in blue, if you can see that, is the low light data. So it goes down between 20 and 23. But the rate of respiration, if you take the pink data, between 20 and 23 goes up. So the net rate of photosynthesis is going down due to low light, and the rate of respiration is going up due to the higher temperature or the increase in temperature. So we've got a lower level of net photosynthesis and we've got higher respiration rates. So that means that actually there's less photosynthates available to build biomass. And if there's less dry biomass, it means that the growth, which is the production of new cells, is going to decrease. The rate of growth is going to decrease over time. So if you guys just have a quick look at the answer. So the growth will decrease. So that's cell division to form new tissue, which is basically dry biomass. The rate of respiration will increase. So we know it goes up. That was the pink data, between 20 and 23 degrees C. Net photosynthesis will be limited by lights because of the clouds, and they block the light at low light levels. So actually the net rate of photosynthesis went down somewhat between these two temperatures in low light. So we'll get less growth of the plant. So a student investigated the rate of anaerobic respiration in yeast. Now we know yeast are unicellular organisms. That means each organism is made of a single cell, but they are eukaryotic. So a yeast cell will have a nucleus with linear DNA. It will have other organelles, including mitochondria. However, we know that yeast cells, in the absence of oxygen, can undergo anaerobic respiration. Now, this will involve glycolysis in the cytoplasm of the yeast cell. So the starting molecule will be glucose, and that will undergo, we could put glycolysis, and that will make pyruvate. And you get 2 of those per glucose, because the pyruvate is a 3-carbon. Now, in the absence of oxygen, the mitochondria are not involved. There's no oxidative phosphorylation. So what happens to the pyruvate? 2 things. In yeast, the pyruvate is decarboxylated to release CO2 gas in the cytoplasm, and there's also a reduction reaction to make ethanol. Now, the ethanol is an alcohol, and that is a 2-carbon molecule. And the purpose of this reduction to ethanol is that NAD, the oxidized version, is regenerated, which allows more glycolysis to continue, because NAD is needed for the reaction of triose phosphate to pyruvate. So NAD, the oxidized version, will go back into glycolysis up here, and it will pick up more hydrogen during the oxidation of triose phosphate to pyruvate. So this anaerobic respiration in yeast cells allows glycolysis to keep going. Now, the scientists put 5 grams of yeast into a glucose solution and placed this in the apparatus shown. So we know 5 grams is going to be really important in some of our calculations a bit later. And the glucose here, we know, is the starting molecule for respiration. And we know it gets phosphorylated to form glucose phosphate. And that's the first part of glycolysis. So the scientist has recorded the total volume of gas collected every 10 minutes for 1 hour. So every 10 minutes, the scientist has measured the volume of CO2 gas. So I would put CO2 gas because that's being produced from anaerobic respiration. And she's basically adding the gases every 10 minutes to get a total volume up to 1 hour. So it says, explain why there was a layer of oil in this investigation, which we can see here in red. So the yeast cells are in the mixture with glucose. Now, what you don't want is oxygen getting to the yeast cells. So the layer of oil was basically to stop the oxygen gas getting to the yeast cells, so we could start to investigate the production of CO2 gas through anaerobic respiration. So the layer of oil is required to prevent oxygen being taken up or entering by simple diffusion the yeast. Now we know oxygen is non-polar, and we actually know it's lipid soluble. So normally it can simply diffuse across phospholipid bilayers, but here it's not being allowed to get near to the yeast cells. So in question B, we're given some data. We've got time in minutes in the left column, and we've got the total volume of gas collected in centimetre cubed. So we know we've got anaerobic respiration with the production of CO2 gas. So that's the gas being collected. Obviously, we've got some ethanol as well. That's not a gas though. And it says, calculate the rates of gas production in centimetre cubed per gram per minute. And that's during the first 40 minutes of the investigation. So if we look at the units then, it's going to be the volume of gas per gram. So we could say per gram of yeast. Now we know in the question we were given 5 grams. So whatever value we're going to calculate, we're going to divide it by 5 to get per gram. And then it says per minute. Now we know the total volume of gas has been collected in 40 minutes. So here we would have to divide by 40 to get to per minute. So if we look at 40 minutes on the table, there's a total, because it says total at the top, 3.1 centimetre cubed of CO2 gas. Divide that by 40 to get to per minute. So we divide by 40 to get to per minute. and divide by 5 to get to per gram of yeast, because it said 5 grams of yeast have been used. So the total volume of CO2 gas collected after 40 minutes was 3.1 centimetre cubed. The volume of CO2 gas per minute divided by 40 is 0.0775 centimetre cubed. But again, that's in 5 grams of yeast, but we need it to per gram of yeast. So if you divide 0.0775 by 5, we're going to get 0.0155 centimetre cubed of gas, CO2 gas, per gram of yeast per minute. So there were 2 marks for 0.0155, or you could have rounded that up to 0.016. And there was 1 mark for the value of 0.0775, but remember that is in 5 grams of yeast, and we need it per gram. So always go to the units in the maths question first, and then you know exactly what it is you need to work out. So suggest why the rates of gas production decreased between 50 and 60 minutes. So if you actually look at the total volume of CO2 gas being produced, there is actually an increase in CO2 gas between 50 and 60 minutes, but the rate of its production is actually lower or slower compared to earlier in the investigation. So we know that glucose is the starting molecule for respiration, and in anaerobic, like we said earlier, we get CO2 gas and ethanol inside yeast cells. So the answer to this could be one of 2 things. It could either be that the starting molecule has run out or has become limiting, because obviously the glucose is being used up, so there's less of it present over time. Or it could be that the ethanol, because that accumulates over time, we know ethanol is toxic to yeast cells in high concentrations. So we could have, and you guys should actually put in all of these points, the glucose decreases over time, so it becomes a limiting factor. Or we could have there's an increase in the ethanol concentration, which we know is toxic to yeast cells. If there's less yeast cells, then there's less respiration taking place. Right, the second point to this slide then says, yeast can also respire aerobically. The student repeated the investigation with a fresh sample of yeast in glucose, but without the oil. So we know now the oxygen can simply diffuse into the yeast cells, and it diffuses into the mitochondria across the 2 membranes, the outer and the inner membranes of the mitochondria. Now we know oxygen is obviously non-polar and it is lipid soluble, so it just diffuses from a higher to a lower concentration into the mitochondria where it gets used up. All other conditions remain the same. So explain, using our knowledge of biology, what would happen to the volume of gas in the syringe if the yeast were only respiring aerobically. So the role of the oxygen then is, as we'll see in a minute, the final electron acceptor. So oxygen gas is being used up inside the mitochondria to form water, but we also know from the balanced chemical equation that CO2 gas is being produced. So if we just write out the balanced chemical equation first, which you should always do, so C6H12O6 is glucose, plus 6 molecules of oxygen will produce 6 molecules of CO2 gas, plus obviously water that's actually formed from the oxygen, plus electrons and protons. So the concentration of oxygen gas being used up is the same as the concentration of CO2 gas being produced. So the volume of gas being used is the same as the volume of gas being produced. Therefore, there's no change in the volume of gas over time. Okay, so it stays the same or no change. Now, just for your notes, CO2 gas we know is produced in the link reaction inside the mitochondria, and it's also produced from the Krebs cycle from the 6 carbon to the 5 carbon and the 5 carbon to the 4 carbon via decarboxylase enzymes. Okay, so 1 mark for saying the volume of CO2 gas remains constant or the same. So the same volume of oxygen is taken up as the final electron acceptor to form H2O versus the CO2 volume or concentration that's being released from aerobic respiration. Okay, respiration produces more ATP per molecule of glucose in the presence of oxygen than it does when oxygen is absent. So explain why. So obviously this question is about what happens to the oxygen inside the mitochondrion. So we said earlier it's the final electron acceptor, and it does pull the electrons through the ETC. Now oxygen plus electrons plus some protons, we could put half there, will form molecular water at the end of the ETC. So this oxygen allows the movement of electrons through the ETC, that activates the proton pumps, that builds the electrochemical gradient, that allows protons to move back through the stored particle from the intermembrane space where it's very acidic to a high concentration of protons back into the matrix, and therefore that allows oxidative phosphorylation. Okay, which makes, as we know, lots of ATP. So there's actually 34 molecules of ATP made via oxidative phosphorylation using the electron transport chain and ATP synthase. So oxygen is the final electron acceptor, and it combines with electrons and protons to form water. So you guys could annotate this in the space above. That allows oxidative phosphorylation to take place, whereby lots of ATP is being produced from ADP and PI. Now, in the absence of oxygen, it also accepted there's no link reaction and no Krebs cycle and no electron transport chain. So there's no oxidative phosphorylation in the absence of oxygen. There would only be glycolysis in the absence of oxygen. Now we know glycolysis makes net of 2 ATP per glucose, which is obviously much smaller than 34 ATP by oxidative phosphorylation. That would be per glucose at the top as well. Okay, the next question then, it says scientists investigated changes in plant biodiversity in different communities after changes caused by humans. They collected data from many published investigations that recorded changes in species richness. So you guys can actually write down what that means. Okay, it's the number of different species in a given area in a given time. Okay, so that's nothing to do with the individual numbers. It's just to do with the number of different species present. You don't need the population numbers of each species. The scientists used data from each investigation to calculate the effect size. The effect size is a measure of the change in species diversity with time. A positive value shows an increase in species richness with time. So if we look at a 0 value here, if it's positive, there's an increase in species richness. Okay, and if it's negative, there's a decrease in species richness. So the mean effect size 0 is the start point. The graph obviously shows the scientists' results in the form in which they were published. The horizontal bars represent plus or minus 2 standard deviations above or below the mean value, which includes just over 95% of the data. So if we look at this data point at the top, that's the mean value, the mean effect size. Here we've got plus 2 standard deviations, and the other bar is minus 2 standard deviations. And it's the same for the next human activity. So the first one was land cleared to be used by humans and then abandoned. The second was maybe a fire. So the next mean value, the effect size, will be for fire. And again, we've got 2 SDs below and 2 standard deviations above the mean, which are these bars here, hopefully you can see. Now, if you look at all the data, the only 2 human activities that the standard deviation bars do not overlap with the 0 value is for the one at the top and the introduction of non-native species. So if you look at the mean value here, and then you've got 2 SD beneath it and 2 SD bars above it. So if I just put circles around here, this one and this one, the SD bars do not overlap with the 0 mean effect size. So therefore we could say there's a significant increase in mean effect size for land cleared and used by humans, and we could say there's a significant decrease in mean effect size for introduction of non-native species. Now, if we look at the fire, we can see the SD bars overlap with the mean effect size, so there's no significant change in effect size. That is the same for grazing animals, and it's the same we can see as well, even though there's a bigger spread of data away from the mean value for the removal of grazing animals, and we can see also for climate change and the effect of pollution, where the SD bars all overlap with the 0 mean effect. So no significant change would encompass all this data in the middle. Okay, so what can you conclude from the data about the effects of human activities on biodiversity? So we know there's a number of different human activities that were given in the information on the left. So we know, first mark point, that it's only the cleared and abandoned land and the introduction of non-native species that makes a significant difference. So we know the first one, there's a significant increase in the mean effect size, and the non-native species, there's a significant decrease in the mean effect size. Now, because the standard deviations, there's plus or minus 2 standard deviations either side of the mean value, and we know they don't overlap with the 0, so the mean value must be more than 2 standard deviations away from the 0 mean value where there's no change. Now, we could also conclude that 4 of the activities increase species richness, which are the top 4, and 3 of the activities actually decrease species richness. So you could even name the activities which increase and name the activities which decrease the species richness. So suggest an explanation for the effect size when non-native species were introduced to communities. So let's have a look. Non-native species. Now, in the information in the question, at the start of the question, it does say that non-native species were introduced. Now, if these non-native species were plants, for example, species of plants, they might outcompete the native species of plants. Now, you know the native species are plants because it gives you this in the information at the start of the question. So if you guys go back and have a quick read through the text, it says the effect on the native species of plant. So if the non-native species that was introduced was a plant, it might outcompete the native species of plant, and that could be for light, it could be for carbon dioxide, it could be for nitrate ions in the soil, for example, it could be for soil water. Now, if the non-native species that was introduced was, for example, a herbivore or an omnivore, well, we know they eat, if they're primary consumers or secondary consumers, they could eat the producer, which is the native species of plant, which could also cause a significant decrease in the species richness of the native species of plants. So the native species could become extinct from the community over time. Now, describe how you would investigate the effect of an invasion of a non-native species of plant. So this time it is talking about the non-native species, that's the one that's being introduced into that area, is a type of plant, species of plant. So we know this is going to compete, like we said at the top, for the native species of plants for light, CO2, nitrates, water, etc. Now, the first thing I would do is to write down, because plants don't move and this is in a given area, we're going to use random sampling. So random sampling is a method in the Investigating Populations booklet to estimate the population number of different species of plants in a given area. So the first thing you're going to do is draw a grid like this over, so draw a grid over a map of the area, You then generate random coordinates with a random number generator like a calculator, and you place the center of each quadrat at each given random coordinate. Now we need a large number of quadrats to make sure the data is representative data, and we can generate a reliable mean value. You could say that you're going to have enough quadrats where if you calculate the mean value every time you make a measurement, you're going to get a stable running mean. So where the new mean value remains pretty constant over time, over more data values. Okay. Now, what you do to get the estimate of population size, you could either do percentage cover. So if you had a quadrat with 100 little squares and you saw that particular species in 38 of the small squares, that would be 38% coverage. Or you could do abundance, where you actually count the numbers of individuals of that particular species of plant. But what you'd have to do is, if you had, say, 30 quadrats and you were doing percentage cover, you'd calculate a mean percentage cover. So you'd add up all the values for percentage cover and divide by 30 to get a mean value. Or if you were doing abundance, so we could say abundance, couldn't we? You would add up all the individual numbers from the 30 quadrats, get a total, and then divide that by 30 to get a mean value. So that's how you generate the mean value for estimating the population size. Now you could also scale up to that given area. So if the quadrat was a meter squared, you'd get a mean value per meter squared, and then you could just scale up a bit. Now, because it says over many years, so this is really, really important. So look at this. So please use the info. Okay, so estimate of population size over many years. So you're going to have to select the same time each year. So it could be maybe August that you select, whatever the month is. And then you repeat the experiment every August for a number of years to see the effect of how population numbers change over this time period. So question D is the effect size, and it tells us that that is calculated using the following set of instructions. And then basically what we're going to do is use our calculator and input the values into the set of instructions to come up with the value at the bottom for effect size. So it says divide the species richness in the last year of the investigation, so that is SR2, by the species richness in the first year of the investigation, so SR1. So if we just follow the set of instructions, starting with number 1 at the top, okay, we know SR2, whatever value that is divided by SR1. And then it says find the natural log of the result of that division. Now, if you look down at the bottom of the information, it says on a calculator, the key for natural log is shown as IN or log E. So you're either going to use the log E button, or it says this symbol, because they mean the same thing. So basically we're going to do the natural log of this first division, which is point number 2. And then it says point number 3, divide the answer that we get at the top by the time between the first and the last year in decades. So this decade is really important. So we know SR2 is 15.3. We know SR1 is 18.2. And we know the time in years is 29 years, but we know that is 2.9 decades. So the last thing we need to do is divide our original division by the time in decades. So let's just write out the working out. I'll do this at the bottom then on the left. So the natural log of 15.3 divided by 18.2 divided by 2.9, because that's the number of years in decades, remember, and that will give us 0.0599. So if you guys got 0.0599, then that's 2 marks. If you demonstrated the equation, which is the natural log of SR2 divided by SR1 divided by the time in decades, that would have got you 1 mark. So there's 1 mark for showing how you would do the working out, and the second mark was for obviously inputting the correct numbers. So if you were a bit confused about log to the base e, it kind of did say at the bottom in the information here that it's just that symbol button on your calculator. So let's try and follow the information as best we can. Okay, the last question then is an AO1 recall question. Obviously you guys need to learn the LDR that occurs at the thylakoid membranes inside chloroplasts, and then obviously the LIR, which is in the stroma of the chloroplast, which is where the Calvin cycle takes place. So it says crops use light energy to produce photosynthetic products. Describe how crop plants use light energy during the light dependent reactions. So light energy we know, so it's blue light and red light, and they are absorbed by chlorophyll. We know green light is reflected by chlorophyll, so that's not going to be used in photosynthesis. Okay. Describe how crop plants use the light energy during the LDR reactions. So here there's 3 main ideas. So photoionization, and that's where chlorophyll absorbs light energy. Electrons become excited to a high energy level and get released to the electron carrier, the first one in the ETC. So the chlorophyll becomes oxidized as it loses the high energy electrons. We've got photolysis. Now that's the light activated splitting of water, and I would always write the equation for that. So water is split into oxygen gas and electrons and protons. Now the electrons are quite low energy, so they replace the high energy electrons that were lost from chlorophyll as they move down the ETC. So water is very important because it actually supplies electrons to the chlorophyll. And then lastly, we've got photophosphorylation. So as the electrons move down the ETC, we know they lose energy. That builds the electrochemical gradient. So actually the protons are being pumped into the thylakoid space. So you get a very acidic thylakoid space with lots and lots of protons, high concentration. The protons can only move back down their concentration gradient back into the stroma through ATP synthase, which is part of the salt particle. And that movement of protons down the electrochemical gradient will supply the energy to phosphorylate ADP into new ATP via ATP synthase. So let's just recall. So let's have a quick look. Excite electrons. Electrons are then released from the chlorophyll molecule and they're accepted by electron carriers. So it's a series of redox reactions as the electrons move from carrier to carrier down the ETC, point 1 and 2. Okay, we know that builds the electrochemical gradients because the energy lost from the electrons is used to activate the proton pumps. Okay, energy is then used, like we said, by ATP synthase to join ADP and PI to form ATP. That's at the end of the electron transport chain in the thylakoid membrane. Water is very, very important because it's split into protons, electrons, and O2 gas, and the electrons replace those that were lost from chlorophyll. Now these are actually low energy electrons from the splitting of water, and they go into chlorophyll. Now obviously when the chlorophyll absorbs more light, these low energy electrons become excited to a higher energy level, and they then get released down to the ETC. So water allows the LDR reactions to proceed. Now, actually at the end of the ETC in the thylakoid membrane, the final electron acceptor is actually NADP, and it's the NADP that pulls the electrons through the ETC. So NADP becomes reduced by electrons and some protons, and it's going to form NADPH. Okay, so in the chloroplast, the final electron acceptor is NADP, and that becomes NADPH. And that NADPH is really important because it supplies the hydrogen in the Calvin cycle, which is needed for the reduction of glycerate phosphate to triose phosphate. Okay, so GP in the stroma would be reduced to triose phosphate using the hydrogen supplied by the reduced NADP. And that was the last question.